Good evening to all. Welcome back to Excel for MRP class. Today we discuss about again microbiology class for MRP preparation. Okay, see here. Our last class we discussed about microbiology introductions and their classifications. Then we discussed about bacteriology. Already we know that what the meaning of microbiology. Microbiology means is nothing but study about microorganisms. It's called as microbiology. Our last class we discussed about bacteria, then as well as immunology. அப்படின்ற ரெண்டு டாபிக் பத்தி டிஸ்கஸ் பண்ணிருப்போம். Today we discuss about virus. Right. Now we study about virus means is called as virology. We study about virus means the type of study called as virology. Here we study about virus morphology, then virus characters, as well as virus classifications, then viral pathogenicity, then virus cultivations method, then identification and investigations. Okay, இத பத்தி தான் நம்ம overall discuss பண்ண போறோம். Now we study about the virus na, the type of study called as virology. Right. First of all, நம்ம எல்லாத்துக்கும் doubt இருக்கும். What the meaning of virus? Virus means is nothing but they also one kind of microorganisms. அதை மட்டும் first understand பண்ணிக்கங்க. Virus means is nothing but they also one kind of microorganism. They are very smaller than bacteria. Bacteria விட சின்னதா இருக்கக்கூடிய ஒரு microorganisms. Okay, the virus is an obligate intracellular parasites. Parasites means already we know that parasites means they also living organisms. They are depending to another organisms for growth and metabolism. Na they called as parasites. Here the virus they are intracellular parasites. The virus not grow independence growth metabolisms. Virus independent growth agadu. They need some living organisms for growth and metabolisms. So they called as intracellular parasites. Right. Idan virus oda structure. The virus they are contain genetic material. The ella microorganisms paathatha vida the virus they are having some special characters. What are the special characters they are having na? The virus they are having genetical material. Genetical material means is nothing but nucleic acid. Nucleic acid na நம்ம already படிச்சிருப்போம். They are classified two types. One is DNA, another one is RNA. Deoxyribonucleic acid, ribonucleic acid னு சொல்லி DNA RNA படிச்சிருப்போம். So the DNA RNA they are having in that virus. இத சுத்தி they are having one coat. The coat made up of protein. So இப்படி எல்லாம் இருந்துச்சுன்னா the kind of microorganisms called as virus. Right. Then now we discuss about virus properties. What are the properties they are having in that virus? Means it's nothing but listen here. First, the virus they are having genetical material. Genetical material means already we know that DNA or RNA. Then as well as some virus they are having one special coat to protect the nucleic acid. The nucleic acid சுத்தி ஒரு extra coat இருக்கும். அப்படி இருந்துச்சுன்னா they called as envelope. Envelope means is nothing but extra cover. Some virus they are having envelope. The kind of virus they called as enveloped virus. Some virus not having enveloped. The kind of virus called as non-enveloped virus. They are different between enveloped virus and non-enveloped virus. Some virus they are having special coat they called as enveloped virus. Some virus do not having envelope they called as non-enveloped virus. இதான் அதோட difference. Then as well as in the nucleic acid சுத்தி one special protein coat is there. The coat made up of capsid. இங்க மார்க் பண்ணிருப்போம் பாருங்க. The special coat they called as capsid. The capsid எதை சுத்தி இருக்கும்னா the capsid ஓட work they are protect the nucleic acid. They are a genetical material. The genetic material ரொம்ப சேஃப்டியா பார்க்கிறது இந்த capsid. So the capsid react as a one productor or depensor. Okay. In the proteins is made up of in the capsids made up of protein. So they called as nucleocapsid protein. அப்படினு சொல்லுவாங்க. Then the envelope made up of lipid and protein. Lipids நால ஆயிருக்கும். Some envelope made up of lipids, some envelope made up of glycoprotein. The glycoprotein envelope mainly used for attachment to host. இதோடைய mechanisms the glycoproteins mainly used for attachment of host. Or host oda poi attach பண்ணிக்கிறதுக்கு they are easily well supported. Okay, they are properties of virus. Okay, then now we discuss about virus characters. What are the virus characters is there? Bacteria, they are having different kind of characters, as well as virus, they are having different kind of characters. Then fungus, they are having different characters. Parasites, they are having different characters. The all microorganisms, they are having individual different characters is there. Then the virus, they are having some special characters. What are the special characters is there? The virus do not grow independently. Taniya adanala grow aga mudiyadu. They need some support. The virus do not having independence growth metabolisms. The virus individual grow aga mudiyadu. They need some support. When we have one SIM card, then as well as we have one mobile phone na, when we add the SIM card to that mobile, appo mattum dhaan nammalaala call panna mudiyum. SIM or thaniyavum phone or thaniya vechirundamna nammalaala call panna mudiyadhu. Same like that, the virus do not having independence growth metabolisms. When that attached to that host, எப்போ ஒரு living host கூட போய் attach ஆகுதோ, that time only they are having the ability to cause the disease. Okay, the virus, they are having the ability to cause the disease, human and animal, then as well as plant. எல்லாத்துக்கும் எல்லா ஹோஸ்டுக்கும் disease காஸ் பண்ணக்கூடிய ability இதுல இருக்கும். Then normal microscope la we do not visible that virus. We want some high power of lens. The high power of lens, they are presented in that electron microscope. Okay, the electron microscope. The electron microscope, they are classified two types, SEM and TEM. Scanning electron microscope, transmit electron microscope. The rendu electron microscope la mattumna we properly visible that virus. Yena the virus size, they are very smaller than bacteria. So we need some electron microscope na, that time only we visible that. Then now we take one virus na, the virus, they are having only one nucleic acid. Don't both. Rendume adula irukadu. Okay, not both. Rendume adula irukadu. They are having only one DNA, only one genetical material. One DNA irukum, illati RNA irukum. Okay, not both. Rendume ore time la irukadu. Then as well as how to reproduce abdina, they are using replications method. Poga poga paakalam. How to the virus they are replicate அப்படின்றதெல்லாம் ஸ்டெப் பை ஸ்டெப்பா போய் பார்க்கலாம். ஸோ இதை ரிப்ளிகேட் பண்றது எப்படி இருக்கும்னா டைவர்சிட்டி மெத்தட்ஸ். தே ஆர் யூசிங் டைவர்சிட்டி மெத்தட்ஸ். இந்த மூலியமா தான் the virus are replicated. Okay, now we discuss about virus structure. இப்பதான் வைரஸ் உடைய மார்பாலஜி பார்க்க போறோம். Now we discuss about virus morphology. This is a virus structure. Okay, காமனா ஒரு வைரஸ் ஸ்ட்ரக்சர் சொல்லியிருக்கோம். Listen here. The virus, they are having genetical material. The red color irukku parunga. They called as genetical material. The genetical material, they are classified two types. One is DNA or RNA. The virus they are having na, the virus la edavadhu or genetical material da irukum. DNA or RNA, not both. Rendume ore time la irukadu. The some virus they are having some special coat. Inga parunga, dark coat is there. Okay, they called as enveloped. The envelope made up of lipid as well as protein. Okay, then in the yellow color, in the nucleic acid sutti, we are having one yellow color coat. The coat made up of capsid. Okay, the capsid. The capsid is mainly used for to product the nucleic acid. So they called as nucleocapsid. They are mainly present in nucleus. The nucleus pakkathula irukanaala to product the nucleic acid. So they called as nucleocapsid. Then in that we are having on the enzymes. The enzyme they mainly used for to product the nucleic acid. Right. Then inga parunga, capsid. The capsid irukle, and the capsids, the some virus they are having the more amount of capsomerase. Capsomerase means is nothing but the capsid or the pods they called as capsomerase. The more kind of pod is there in that capsids. So in the capsids they are different between some sila virus ku maaride irukum. So here we are having the capsid complex. Complex means poly, adhigama more, adhigama adula irukum. Then now we discuss about virus type. Virus they are classified three types. Inga four types irukum. But the virus, they are classified 3 types. One is spherical virus, another one is helical virus, another one is complex virus. ஏன் எக்ஸ்ட்ரா ஒரு வைரஸ் கொண்டு இந்த இடத்துல வச்சுட்டாங்கன்னா they are structures like இப்படிதான் ஒரு மாடல்ல இருக்கும், but they are coming under helical structure. They are coming under helical structure, but அதோட ஸ்ட்ரக்சர் லைக் டைமண்ட் மாதிரி இருக்கனால they are coming under polyhedral virus அப்படின்னு சொல்லி they are classified. So totally the virus, they are classified 3 types. One is spherical shaped virus, another one is helical shaped virus, another one is complex virus. Then additionally we are having another one types they called as polyhedral virus. Okay, for example spherical virus ku example coronavirus as well as adenovirus, some virus they are having in that spherical shaped. Then helical shape na tobacco mosaic virus, then influenza virus, then some virus they are coming under helical virus. Same adhe mari complex virus T3 bacteriophage as well as T4 bacteriophage, the kind of bacteria virus they are coming under complex. Then inge eduthomna polyhedral they also come virus is there. Example pox virus they are coming under polyhedral virus. Indha mari sila virus they are coming under the kind of types. In the types kila vandrum. Once again I repeat that the virus they are classified 3 types. One is spherical, helical, complex. Additionally we have another one type they called as polyhedral structure. Polyhedral structure means it's nothing but they are having same character of helical structure, but they are having appearance for helical mari or appearance adula irukum. So they called as polyhedral structure. Okay, now we discuss about virus replications. How to the virus they are replicate. Virus eppadi develop agudhu, how to replicate. Replicate means or virus eppadi vandu cultivate ஆகி next one அதோடைய numbers எப்படி increase பண்ணுது. Already we know that virus characters, the virus do not having independence growth metabolisms. The virus they are need some support for growth and metabolisms. When that virus they are attached to that host, எப்போ ஒரு virus ஒரு host கூட போய் attachment ஆகுதோ that time only the virus they are activated. So இந்த yellow color இருக்கிறது they are a bacterial cell, the blue color இருக்கிறது virus. When that virus they are attached at, they called as bacteriophage. The bacteriophage form ஆயிட்டு the virus they are attached to that host. இங்க host யாரு bacterial cell. So virus they are attached to that living cell. Bacteria குள்ள attach ஆயிருச்சு. So they are activated. Then the virus they are do the process of penetration. First one attachment, second one penetration. Okay, penetration, attachment, penetration. Here the nucleic acid they are easily penetrate, then they are mingled with plasmid. The bacterial plasmid kuda they are mingled. Then next process biosynthesis. Biosynthesis na here the virus part is there. They call as head, fiber, then tile, genetic material. இந்த மாதிரி இருக்கு இல்லையா? So they are having one part. The part எல்லாமே synthesis ஆகும். So the stage called as biosynthesis. Okay, next stage assembly. So here the virus they are developed completed virus. இங்கே the first one attachment, penetration, biosynthesis. The virus they are attached to that bacterial cell, then they are penetrate to that genetical material, then the genetical material they are attached to that plasmid. Again the third process we occurring biosynthesis process. Then after that the virus they are attached to that full completed virus ஆயிருச்சு. Last stage they are blast and exit. The last stage the virus they are come out from that the bacterial cell wall. Bacterial cell wall irundhu the more virus they are come out. Then again they are easily attached to that bacterial virus, bacterial cell. இப்படியே இந்த replications போயிட்டே இருக்கும். Once again I repeat that viral replications, first stage attachment, penetration, biosynthesis, assembly, blast or release. Okay, so singular in the virus, here we occurring more virus like 5 or 10, not only 4, இங்க நிறைய வர அளவுக்கு the virus they are developed in that replications method. Okay. Now we discuss about virus cultivations. How to cultivate that virus. எப்படி வைரஸை cultivate பண்ணலாம்? Why we want to cultivate the virus na? When we cultivate the virus, that time only we easily understand the virus characters. அப்போ மட்டும்தான் வைரஸோட கேரக்டர் நம்மளால ஈஸியா அண்டர்ஸ்டாண்ட் பண்ணிட்டு then we treat that virus. Okay, the virus cultivations இதுக்கு முன்னாடி நம்ம சொன்ன மாதிரி ஒரு இம்பார்ட்டன்டான மேட்டர் நம்ம மைண்ட்ல வச்சுட்டே இருக்கணும். The virus do not having independence growth metabolisms. The virus they need some support for growth. So living cells இருந்தா மட்டும்தான் the virus will grow. Okay, cultivations method. Cultivations method they are classified 3 types. One is animal inoculations, embryonated egg culture method, then third one is cell culture methods. So animal cell culture method na here also having living organisms. Embryonated egg na ada vecha koli mutta. So idella kuda the living cells they are presented. Then cell culture group of cells called as tissue. Inge tissue cut பண்ணுவோம். So they called as culturing method. So in the மூணு method use பண்ணி we easily cultivate the virus. The virus cultivation they are classified 3 types. One is animal inoculations, embryonated egg method, cell culturing methods. Okay, this is a animal cell culturing method. This is our embryonated egg culturing methods. They mainly use it for cultivation of virus. Okay, இங்க ஒரு மைஸ் இமேஜ் கொடுத்திருப்போம். Okay, in that genetical laboratories or more research laboratory we are using mice like some animals மட்டும் யூஸ் பண்ணுவோம் for they called as laboratory animal. Why we choose that mice for our testing na? They are having 20 pair chromosomes like a human chromosomes மாதிரியே இது இருக்கும். So we choose the mice for our research. அதுக்காக தான் இந்த mice எல்லாம் யூஸ் பண்ணிட்டு இருக்கோம். Okay, then we cultivate the virus samples. Virus samples வந்து எங்க இருந்து நம்ம டெஸ்ட் கொடுக்குமோ அதை எடுத்துட்டு then we inject to that animal. இந்த animal கொடுத்துட்டு then we keep it the incubations. Incubation வச்சு பார்த்தோம்னா we identify the characters. இதோட கேரக்டர்ஸ் மாறி இருக்கும். இது பெரிய ரிசர்ச். ஸோ நீங்க ஜென்ரலா தெரிஞ்சுக்கிட்டீங்கன்னா போதும். ஸோ இதோட கேரக்டர்ஸை வச்சுட்டு ஈஸிலி ஐடென்டிஃபை த வைரஸ் they are presented or not. Okay, next embryonated egg method. Embryonated egg method means already we know that we take the 6 to 8 days embryonated egg. ஒரு கரு உருவான 6 ல இருந்து 8 நாள் ஆன ஒரு எக் எடுத்துட்டு then we open that place. இந்த பிளேஸை அழகா ட்ரில் பண்ணி ஓபன் பண்ணிட்டு then we inoculate the sample to that the holes. இந்த holes வழியா we properly inoculate that. Then we pack with gelatin. Gelatin வச்சு இந்த இடத்த பேக் பண்ணிடுவோம். பேக் பண்ணிட்டு then keep it under the incubations up to 48 hours. After 48 hours we take that same the embryonated egg then we break that. இதை நம்ம செக் பண்ணி பார்த்தோம்னா here we occurring black hues. அப்படியே black கலரா வந்து இந்த சிக்கெல்லாம் இறந்து போயிருக்கும். இந்த இடத்துல black color ஆயிருக்கும். They called as virus. Okay, இதை எடுத்துட்டு போய் டெஸ்ட் பண்ணோம்னா what are the virus they are presented அப்படின்றத we used by electron microscope. Electron microscope ல ஈஸியா நம்ம பார்த்துக்கலாம். Okay, once again I repeat that virus cultivations la very important method. One is animal cell culture method, another one is embryonated egg method. 6 to 8 days embryonated egg எடுத்துட்டு then we inoculate the sample. Then after 48 hours incubations we properly identify the bacteria virus samples. என்ன samples அதுக்குள்ள கொடுத்தோன்றத நம்ம கல்டிவேட் பண்ணி ஈஸியா அதோட கேரக்டர்ஸ் ஐடென்டிஃபை பண்ணிக்கலாம். Okay, okay, then now we discuss virus now we discuss about virus morphology, virus classifications, then virus structure. But now we go for virus one by one over virus பார்த்துருப்போம். Now we discuss about HIV. HIV na already we know that எல்லாத்துக்கும் நிறைய டைம் இந்த பேரை கேள்விப்பட்டிருப்போம். Human immunodeficiency virus. What are the difference between HIV and AIDS? இன்னும் நிறைய பேத்துக்கும் அந்த டிஃபரன்ஸ் தெரியாம இருக்கும். The HIV means is nothing but they are a virus name. The virus they are cause the disease to human na the disease name AIDS. Okay, HIV means human immunodeficiency virus. Okay, now we discuss about structure of HIV virus. HIV virus உடைய structure இதல பாப்போம். Okay, they are having single stranded. Already we know that the genetical material they are classified 2 types. One is DNA and RNA. எல்லாத்துக்கும் தெரிஞ்சிருக்கும். The DNA, they are having double-stranded helical structure. RNA means they are having single-stranded structure. Okay, here we are having single-stranded. So they called as RNA. So இது உங்களுக்கு இதுல கேட்கலாம் கொஸ்டின்ஸ்ல. The HIV virus, they are having DNA or RNA. The HIV virus, they are having viral RNA genome, single-stranded RNA. Then as well as they do not having envelope. Sorry, they are having envelope. The envelope made up of phosphate or plus lipid bonds. இந்த இடத்துல ரெட் கலரா இருக்கிறது envelope. The envelope made up of phosphate bond. Then as well as they are having glycoprotein. The glycoproteins mainly used for attachment to that host. Or host கூட போய் attachment ஆகிறதுக்கு they are well supported. Okay, now one person they are infected by HIV na, what are the symptoms they occurred அப்படின்னா the kind of symptoms fever, then அதுக்கப்புறம் night sweating, இதெல்லாம் ரொம்ப காமனானது, mouth ulcer, so chest pain, muscles இந்த மாதிரி பெயின், இந்த மாதிரி அடிக்கடி நிறைய symptoms வந்துகிட்டே இருக்கும். So they are a common symptoms for HIV. When that virus they are entered அப்படின்னா first of all they are causing symptoms for fever. First fever cause பண்ணோம், then அதுக்கப்புறம் one by one vomiting, then as well as muscles pains. What are the laboratory test we are used for HIV? Means we are எல்லாமே பேர் கேள்விப்பட்டிருப்போம். ELISA, ELISA. The ELISA method is commonly used for identification of viral disease. The ELISA one not for HIV, HIV க்கு மட்டும் கிடையாது. They are mainly used for all kind of viral disease. The HIV mainly used for identification of disease. ELISA means enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. They are having 4 types. In the ELISA la மொத்தம் 4 types இருக்கு. Direct ELISA, indirect ELISA, sandwich ELISA, then competitive ELISA. So totally they are having 4 types. In the 4 type ELISA la enna methods நடக்கணும்னா, when we are using that ELISA method, that time we identify the antigen antibody reactions. எப்போ ELISA test use பண்றோமோ that time easily identify the antigen antibody reactions. Then we are using the Western blotting method as well as PCR method, polymerase chain reactions. So இந்த மெத்தட்லாம் யூஸ் பண்ணி we easily diagnostic the HIV க்கு diagnostics பண்ணலாம். Then antibody testing. What are the antibodies we are having? Then what are the antibodies they are infected by the HIV pathogen அப்படின்றத நம்ம ஐடென்டிஃபை பண்ணலாம். As well as NAT, nucleic acid test. NAT, the nucleic acid test. The test they are mainly used for HIV diagnostics. Okay, next we discuss about hepatitis. First we discuss about HIV, now we discuss about hepatitis. Hepatitis means is nothing but we occurring inflammations. We occurring inflammations of liver is known as hepatitis. Liver la we occurring some inflammation na, they called as hepatitis. What are the factors we are having for hepatitis na? Autoimmune disease. For last class we discussed autoimmune disease. Then some tissue problems also there. Autoimmune disease மூலமா வரலாம், tissue problem மூலமா வரலாம். Sometimes our cells damage, cells கூட damage ஆனாலும் கூட we occurring hepatitis, inflammation of liver கூட வரலாம். So they are a chronic disease. இது ஒரு நாள்பட்ட டிசீஸா தான் இருக்கும். What are the symptoms we occurred? Already we know that abdomen pain, chest pain, jaundice, the dark urine, they are a common symptoms for viral disease. The fever, இது எல்லாமே வந்துகிட்டே தான் இருக்கும். Vomitings, இது எல்லாமே தான் வந்துட்டே இருக்கும். So they are a common symptoms for viral disease. Then the hepatitis, they are classified 5 types. One is hepatitis A virus, hepatitis B virus, hepatitis C virus, then hepatitis D virus and E virus. They are the hepatitis virus, they are classified 5 types: A virus, B virus, C virus, D virus E virus. So totally they are having 5 types. Okay. Then இங்க தான் நீங்க கரெக்ட்டா பார்த்துட்டு இருக்கணும். Some virus they are having DNA. The hepatitis B virus they are having DNA genome. Then hepatitis A virus they are having RNA genome. The hepatitis E virus they are having RNA genome. Then hepatitis D virus they are having RNA genome. Then hepatitis D virus they are having DNA genome. So they are a different between hepatitis A, B, C, D, E. இதான் அதோடைய கிளாசிபிகேஷன்ஸ். பட் அதை தவிர they are having same. எப்படி என்ன what are the properties, main properties is there? அந்த ப்ராப்பர்ட்டிஸ் எல்லாம் இதுக்குள்ளேயும் இருக்கும். They are having enveloped, they are having membrane, then as well as they are having capsids, they are having the lipid envelope. Then இங்க பாருங்க இங்கேயும் capsids இருக்கும். But they are having different kind of genome. The hepatitis B virus they are having DNA, deoxyribonucleic acid. The hepatitis A virus they are having RNA, ribonucleic acid. The hepatitis E virus they are having RNA. So இதுதான் அதோட டிஃபரன்ஸ். இதை மட்டும் கொஞ்சம் ஈஸியா அண்டர்ஸ்டாண்ட் பண்ணிட்டா இன்கேஸ் ஒரு கொஸ்டின்ல கேட்டா நம்ம ஆன்சர் பண்ற மாதிரி இருந்துக்கணும். So the hepatitis A virus, hepatitis B virus, C virus, D virus, E virus. So totally they are having 5 types. So they are having different kind of genome. இதை மட்டும் கொஞ்சம் அண்டர்ஸ்டாண்ட் பண்ணிக்கணும். Okay. Next we discuss about what are the diagnostics we are used for hepatitis. Same. The common viral diagnostics we are used for identifications or investigation of virus. First, analysis of genetical material. The analysis of genetical material. What are the genetical material they are presented? The DNA or RNA. Then antibodies they are infected by that pathogen. They are a first diagnostics. Second one, what are the antibodies they are involved? அப்படின்றத பார்க்கணும். Third one, the liver function test. Liver எப்படி function ஆகிட்டு இருக்கு? They are having any critical conditionல தான் இருக்கா? They are having normal condition அப்படின்றத நம்ம அண்டர்ஸ்டாண்ட் பண்ணணும். Then அதுக்கப்புறம் ஹிமினோகிராம் டெஸ்ட் பண்ணலாம். Then biochemical blood test. Then complete blood test. They are a very important diagnostics for hepatitis. So இதெல்லாம் பண்ணோம்னா we easily identify, then easily find out the hepatitis. One person infected by some fever like that, இந்த மாதிரி அவருக்கு நிறைய fever வந்துட்டே இருக்கு அப்படின்னா போய் ஹாஸ்பிட்டல்ல செக் பண்ணி பார்த்தோம்னா the liver they are having some inflammation na, then we go for the testers. Okay, the analysis of genetical material as well as antibiotics test, then liver function test, the complete blood count test, the biochemical blood test இதெல்லாம் பண்ணி பார்த்தோம்னா easily we identify the hepatitis. In case we have hepatitis disease na, we easily identify the hepatitis type, which kind of virus they are having, hepatitis A virus or B virus or C virus or D virus அப்படின்னு சொல்லிட்டு then we go for treatment. Okay, next we discuss about poliomyelitis. Physiotherapist இது உங்களுக்கான ஒரு ரொம்ப இம்பார்ட்டன்டான ஒன்று தான். போலியோ நிறைய பேஷண்ட்டை நீங்க பார்த்துட்டே இருப்பீங்க, போலியோனால இன்ஃபெக்ட் ஆகி வந்தவங்கள. ஸோ த போலியோ வர்றதுக்கு காரணம் ஒன் கைண்ட் ஆஃப் வைரஸ், த நேம் ஆஸ் போலியோ, த poliomyelitis virus. So we have poliovirus structures. Here the poliovirus they also having genetical material RNA. So they do not having envelope. இதுல இப்படி ஒரு கவர் கிடையாது. They are having only membrane. The poliovirus do not having enveloped. They are having membrane. Then they are having capsid seal. பாருங்க ஒரு சீல் மட்டும் இருக்கும். They are having capsid. They don't have special coat. The special coat called as envelope. They don't have envelope. So they called as non-enveloped virus. Non-enveloped virus. Okay. Then what are the symptoms we occurred? நிறைய போலியோ பேஷண்ட்டை பார்த்துருப்பீங்க. ஸோ அவங்க வந்து இந்த ஃபீவர் இருக்கும், ஹெட்ஏக் இருக்கும், வாமிட்டிங் இருக்கும், தென் as well as டயரியா போயிட்டே இருப்பாங்க, அப்படியே நெக்கெல்லாம் அப்படியே ஸ்டிஃப்னஸ் ஆகி அப்படியே இருக்கும். தென் சம்டைம்ஸ் ஃபீவர் கூட ஹையா இருக்கும். பேரலைசிஸ் ஒரு சைடா அப்படியே பேரலைஸ் ஆகிட்டு காலெல்லாம் வீங்கி மசில்ஸ் எல்லாம் ரொம்ப வீக்கா இருந்துச்சு அப்படின்னா சம் பீப்புள்ஸ் 95% the persons they are infected by polio. ஈஸியா இந்த மாதிரி சிம்டம்ஸ்லாம் இருந்து மசில்ஸ் எல்லாம் வீக்கா இருந்துச்சுன்னா we easily identify persons they are infected by polio, right? Then what are the laboratory diagnostics we are used அப்படின்னா medical history, அவங்களுடைய மெடிக்கல் ஹிஸ்டரியை செக் பண்ணி பார்க்கலாம். தென் பிசிக்கல் எக்ஸாமினேஷன்ஸ், பிசிக்கல் எக்ஸாமினேஷன்னா அவங்களை நடக்க வச்சோ இல்லை அவங்களுடைய மசில்ஸ் எப்படி இருக்குன்னு அதை நீங்க உங்க எக்ஸசைஸ் மூலியமாக பார்க்கலாம். தென் அதுக்கப்புறம் ஏதாவது பேரலிசிஸ் இருக்கா அப்படின்னு சொல்றதை நீங்க செக் பண்ணலாம். வாட் ஆர் த laboratory diagnostics we are used அப்படின்னா CFS, the cerebral spinal fluid examination பண்ணலாம். அடுத்து ஸ்வாப்பிங் டெஸ்ட், பிளட் டெஸ்ட், தென் ஃபிங்கர் பிரிண்டிங் போலியோ வைரஸ். போலியோ வைரஸ் இன்ஃபெக்ட் ஆனவங்களுடைய ஃபிங்கர் பிரிண்ட் எடுத்து வாட் ஆர் த வைரஸ் they are having அப்படின்றத we identify some methods. So they called as finger printing polio virus methods. இவ்வளவு மெத்தட் யூஸ் பண்ணி we identify the polio virus. Okay, the poliomyelitis they are a non-enveloped virus. They are having only membrane. Then as well as they are having RNA. Then the glycoproteins, they also having glycoprotein. The glycoproteins mainly used for attachment of host. எல்லா வைரஸ்லயும் இந்த glycoproteins they are presented. They are main supported for attachment that host. Okay, next we discuss about MMR. நிறைய பேர் கேள்விப்பட்டிருப்போம். What the meaning of MMR? MMR vaccine சொல்லி கேள்விப்பட்டிருப்போம். M means measles, another one means mumps, last one R means rubella. So MMR na measles, mumps, rubella. They are three kind of differentiated virus. இது மூணுமே ஒரு டிஃப்ரெண்டான வைரஸ் தான், பட் the characters is same. இதோட கேரக்டர்ஸ் எல்லாம் லைக் சேமா இருக்கும். ஸோ இது எல்லாத்தையும் ஒரு காம்போவா மாத்தி we called as MMR virus, measles, mumps, rubella. What are the difference between measles and mumps and rubella? Na they are structures. Okay, okay, see here the measles they are having nucleocapsids. They are having single stranded RNA. Then இதை சுத்தி they are having capsids, so they called as nucleocapsids. The capsids they are presented in near of nucleic acid. They are product the nucleic acid, so they called as nucleocapsid. The capsids they are present near of nucleic acid, so அதனால they called as nucleocapsids. So இதுதான் structure of measles. Then structure of mumps. Mumps எப்படி இருக்கும்னா they also having RNA. இதுலயும் single stranded RNA இருக்கும். இங்க பாத்தீங்கன்னா தெரியும் RNA they are having. Then they also having nucleocapsids. Then the proteins they are classified. Here M protein is there. Here large protein is there. Then matrix protein is there. Here F protein is there. So they இதான் அதோடைய differentiate. Then as well as rubella. Rubella they are having glycoprotein. இங்க large protein, F protein, இங்க glycoprotein. The glycoproteins, F proteins, then the large proteins they are mainly supported for attachment. Then here the rubella virus they are having icosahedral structures. அதான் நம்ம பார்த்தோம் இல்லையா, the polyhedral structures like that, like a diamond structureல இருக்கும்னு சொல்லி இங்க பாருங்க. Spherical structure na they are having round. Here they are having like a diamond structure, so they called as icosahedral structure. Then the rubella they also having RNA single stranded polymerase structures. Then additionally they are having lipid bilayer membranes. So they are different between mumps, rubella. So they are having the characters they are same. The character ரொம்ப சேமா தான் இருக்கும். So they called as MMR virus. Okay, what are the symptoms we occurred for measles and mumps and rubella? Means first of all, now we infected by measles அப்படின்னா we occurring running nose. காமனா நம்ம ஆல்ரெடி சொன்ன மாதிரி தான் the common viral symptoms they are running nose, then hitching, then fever. Sometimes some rashes is there, some red dots we occurred in our skin. Same like that, the mumps. Mumps vanda kuda fever, then rashes, then as well as fever, then headache, vomitings. They are common symptoms for viral disease. Then as well as rubella. The rubella called as German measles. They are having same like a character of measles. So they known as German measles. The rubella known as German measles. So they are mainly infect to the pregnant ladies. There are pregnant ladies that affect the very critical stage for the pregnant ladies. The ladies are romba critical situation. Sometimes abort they are having more chance for rubella. The pregnant ladies, they are infected by rubella. They are very, very critical. Okay. So they are a MMR. Measles, mumps, rubella. So what are the treatment is there? What are the diagnostics is there? Abdina? Okay. PCR, polymerase chain reactions, as well as ELISA method we also used. Then IgG. What the meaning of IgG? Our last class we discussed about that antibodies. IgG, immunoglobulin gamma, immunoglobulin mu, immunoglobulin expand. So they are antibodies. Here we identify the antibodies. Which kind of antibodies they are involved in the disease? Then sample collections. Then PCR, polymer chain reactions. Here we collect the sample from that throat swapping test. Every corona samples we are using PCR methods. Then serum, we collect the blood. Then we do the process of counting, blood counting. We easily identify the kind of virus, measles, mumps, rubella. Here, now, available MMR vaccines. The babies, they are using MMR vaccines in the interval period. Correct interval period, the people do not infected by measles and mumps and rubella. Okay. Then rabies. Next, we discuss about rabies virus. Rabies virus, now already we know that the rabies is one kind of animal virus. The virus, they are mainly infect to pets. நம்ம வீட்ல ஏதாவது ஒரு பெட் வளர்த்துட்டு இருப்போம். அது டாக் ஆர் கேட், ஏதாவது நம்ம வீட்ல ஒரு பெட் வச்சிருப்போம். அந்த பெட்டோட சலைவால the rabies they are presented. The pet, dog or cat, sometimes human, they also infected by rubella. Saliva vandute irukum. The saliva, the more kind of rabies they are presented. The human or animal, they are infected by rabies na, ivanga yara orthara bite panna, the virus they are easily transmitted to that host, and the host ku maarirum. So they are rabies virus structures. They are having envelope, they are having L protein, and as well as they are having P protein. Then here they are having RNA. Okay. Then M protein is there, N protein, G protein, glycoprotein. So they are having different kind of protein. They are presented in that outer layer. Then as well as they are having enveloped. So they called as enveloped virus. So that is a symptoms. Enna symptoms varum appadina when that pets, they are infected pets, they are bite to human or edha yara bite pannalum அவங்களுக்கு என்ன ஆகும் அப்படின்னா சேம் வைரல் இன்ஃபெக்ஷன் மாதிரி ஒரு ஃபீவர், ஹெட்டேக், வாமிட்டிங், டயேரியா, அந்த சிம்டம்ஸ் எல்லாம் வி அக்கர்ட். as well as சலைவா செக்ரிகேட் ஆகிட்டே இருக்கும். தென் as well as அவங்களுக்கு வந்து ஒரு இச்சிங் இருந்துகிட்டே இருக்கும். பாடி ஃபுல்லா ஒரு இச்சிங், இந்த மாதிரி சென்சேஷன் இருந்துச்சுன்னா easily identify the persons they are infected by some viral disease. Then we go for next treatment. இப்போ நிறைய வேக்சின்ஸ் இருக்கு for rabies. நாய் கடிச்சிச்சுன்னு சொன்னனாலே சேம் லைக் இன்டர்வல் பீரியட் 3 days, 18 days, 21 days அப்படின்னு சொல்லிட்டு we are using some vaccines. நிறைய பேரை வந்து நாயெல்லாம் வீட்டுல குட்டி குட்டி நாயெல்லாம் வளர்த்துறப்போம், பைட் பண்ணியிருக்கோம். So that time we are using interval period rabiesல we are using that vaccines. So அந்த vaccines எடுத்துக்கிட்டோம்னா we control that from rabies. rabiesல இருந்து நம்ம கண்ட்ரோல் பண்ணிக்கலாம். எல்லா பெட்டுமே rabiesனால இன்ஃபெக்ட் ஆகுமா? No. Some kind of pets, they are infected by rabies. Not only pet, human, they also infected by rabies. யாராவது இப்போ என் ஃப்ரெண்ட் இருக்கான், பக்கத்தில் கடிக்கிறான், விளையாடுறான் அப்படின்னா சம்டைம்ஸ் அவருக்கு ஏதாவது ரேபிஸ் டிசீஸ் இருந்துச்சுன்னா டெஃபினெட்லி த பர்சன்ஸ் they also infected by rabies infections. Okay, next we discuss about herpes simplex virus. Okay, herpes simplex virus na they are causing a disease in that oral route. The herpes enna pannona human oda oral route la they are causing a disease. The oral route la disease cause pannum. Okay, inga parunga, the herpes simplex virus they are causing a disease in that oral route. Herpes simplex virus, that is a structure of herpes virus. They also having glycoprotein. They are enveloped virus. They are having one special coat. They are having one special coat. They called as enveloped. Then the herpes simplex virus, they are having double stranded DNA structure. Angela RNA in pathom. The herpes they are having double stranded. DS means double stranded DNA, deoxyl ribonucleic acid. Zoom pani pathina here they are having double stranded DNA structure is there. Okay, then as well as they are having capsids. So the capsids they also product the nucleic acid, so they called as nucleocapsids. So the herpes simplex virus they are cause the disease to human na, mainly the human they are occurring oral infections. Okay, next we discuss about influenza virus. Influenza virus na mostly the human they are infected by influenza. Influenza nala human they are mostly infected. The influenza virus eduthonna they also having RNA polymerase, single stranded RNA polymerase. They also having lipid envelope. Then as well as they are having they also having protein envelope. Rendu envelope idukkulla irukum. What are the symptoms we occur? Same like that. நம்ம காமனா சொன்ன மாதிரி fever, running nose, dry cough, then diarrhea, so vomiting, headache, fever, இதெல்லாமே they are common symptoms for viral disease. Then what are the diagnostics we are used for flu virus? The influenza virus known as flu, so they called as influenza virus. So what are the diagnostics we are used for identification of influenza na? First, the rapid influenza diagnostic test. One method is there. Idala use panni what are the virus they are presented அப்படின்றத கண்டுபிடிக்கலாம். Then PCR, RT-PCR method. Then serological test, antigen antibody reactions. Then virus cultivations method. When we using virus cultivations method na, we easily identify the virus characters. So we are using that virus cultivations method. Then நமக்கு ஆல்ரெடி தெரிஞ்ச மாதிரி NAT, nucleic acid test. Next one, ELISA, enzyme linked immunosorbent assay. The test mainly used for identification of influenza virus. Okay. Next, we discuss about pox virus. Already we know that the pox they are having different kinds is there. Smallpox, monkeypox, chickenpox. The more kind of pox virus is there. They are coming under pox viridae. They are coming under pox viridae. அப்படின்ற ஒரு family கீழ வரும். The pox virus they are having DNA. Okay, then as well as they are having enveloped, lipid enveloped. Then they are having capsids. The capsids they are product the nucleic acid, so they called as nucleocapsid. So what are the symptoms we occurred அப்படின்னா இந்த மாதிரி red color dots we occurring in our skin. Red color dots and rashes la we occurring in our skin. Then what are the laboratory diagnostics we are used for pox virus அப்படின்னா rash examinations. We occurring some dots அப்படின்னா we are using examinations. What are the kind of rash we occurred அப்படின்றத கண்டுபிடிக்கலாம். Then blood test. Any virus they are presented in our blood or not, அது virus or bacteria or fungus அப்படின்றத we are used by blood test. இது மூலமா நம்ம ஈஸியா ஐடென்டிஃபை பண்ணிக்கலாம். Then viral culturing method. We are collect the sample from that patient, then we inoculate the culturing method. Already we know that inoculation methods, animal cell culturing method as well as embryonated egg method. So அதுக்குள்ள நம்ம culturing பண்ணி easily we identify that. Okay, the last one, T4 bacteriophage. Bacteriophage means already we know that they called as bacteriophage. எதனால இத bacteriophage அப்படின்னு சொல்றோம்னா they are having complex structures. The virus they are having complex structure. இங்க பாத்தீங்கன்னா ஆல்ரெடி நாங்க சொல்லியிருந்தேன். They called as head, they called as tail, they called as cooler, then they called as tail fibers. So இப்படி இந்த ஸ்ட்ரக்சர்ல இருந்துச்சுன்னா they called as complex virus. Example, T4 bacteriophage. Why we called as bacteriophage na? இது ஒரு பார்ட் தான். இங்க தான் இருக்கும் ஜெனெட்டிக்கல் மெட்டீரியல். When that virus attached to that bacteria, they called as bacteriophage. எப்ப போய் பாக்டீரியாக்குள்ள அட்டாச்மெண்ட் ஆகுதோ they called as bacteriophage. Okay, so இதுதான் அதோடைய disease causing methods. இது மூலமா they are causing mostly T4 bacteriophage, they are causing infection to plant, vegetables, இந்த மாதிரி இடத்துல தான் poultry, இந்த மாதிரி இடத்துல தான் bacteriophage உடைய infection they are very large. Okay, now we complete that session. Now we go for questions. So கொஞ்சம் கவனிச்சீங்கன்னா இப்ப நம்ம அதுக்கு முன்னாடி என்னென்ன பார்த்தோன்றதை நம்ம ஈஸியா அண்டர்ஸ்டாண்ட் பண்ண முடியும். So listen here. Okay, which of the following is a characteristics of virus? Virus oda characters idhula enna? They have both DNA and RNA. Virus la DNA RNA rendume irukuma. They can reproduce independence of a host cell. Or host cell kuliye the reproduction aikuma. They lack cellular structures. They are classified the prokaryotics. Idhula edu correct answer? Guess பண்ணுங்க. Okay, the correct answer is C. They lack cellular structures. So already we know that they are a intracellular parasites. அப்படிதான் நம்ம இடத்துல first classல சொல்லியிருந்தோம். So the virus don't have DNA and RNA. ரெண்டுமே ஒரே டைம்ல இருக்காது. Then do not having independence growth metabolisms. Then they are classified as a prokaryotic. இது prokaryoticலயும் வராது. So they known as cellular intracellular parasites. So இதான் அதோடைய correct answer. Okay, next we go for next question. What is the main purpose of capsid in that virus? Capsid oda main role enna? Energy production to product the viral nucleic acid, protein synthesis, host cell recognitions. இதோடைய application என்ன? இதோடைய purpose என்ன? Capsid oda purpose என்ன? Okay, the correct answer to protecting the virus nucleic acid. Already நான் சொல்லிட்டே இருந்தேன். The nucleic acid இருக்கும் அதை சுத்தி the capsid they are presented. The capsids mainly protect the nucleic acid. So they called as nucleocapsid. So they are correct answer is protecting the viral nucleic acid. Our next question. Which of the following is a single standard RNA virus? இதுல எது single standard RNA virus? Herpes simplex virus, influenza virus, adenovirus, smallpox virus. Which one virus they are having single standard RNA virus? So நிறைய பேர் கரெக்டான ஆன்சர்ஸ் பண்ணிட்டு இருக்கீங்க. The answer is influenza virus. The influenza virus they are having single standard RNA virus. Okay, our next question. which of the following is not an enveloped virus? இதுல எது enveloped virus அப்படின்னு கேக்குறாங்க. இன்ஃப்ளூயன்சா வைரஸ், தென் நாட் enveloped வைரஸ். இதுல நாட் enveloped இல்லாத வைரஸ் இது. இன்ஃப்ளூயன்சா வைரஸ், ஹெப்படைட்டிஸ் சி வைரஸ், போலியோ வைரஸ், ஹெர்பஸ் சிம்ப்ளக்ஸ் வைரஸ். which virus is a non-enveloped virus? இதுல எது non-enveloped virus? which one is the not an enveloped virus? The answer is polio virus. Polio virus சொல்லும் போதே சொன்னேன். The virus not having enveloped, so they called as non-enveloped virus. So இதான் அதோடைய correct answer. Next question. Which viral structure is the primary responsible for attachment to host cell? ஒரு வைரஸ் உடைய ப்ராப்பர்ட்டிஸ்ல which one is the primary responsible for attachment to the host cell? Capsida. வெரி குட். நிறைய பேர் நல்லா ஆன்சர் பண்ணிட்டு இருக்கீங்க. envelope glycoprotein. The glycoproteins, the main purpose for attachment to that host ஓட அட்டாச்மெண்ட்டுக்கு தான் they are well supported. Our next question. Which of the following virus is known to case of latent infections? இது latent infection காஸ் பண்ணும். இன்ஃப்ளூயன்சா வைரஸ், herpes simplex virus, the rotavirus, then hepatitis A virus. The latent infection cause பண்ற வைரஸ் என்னது? நிறைய பேர் கரெக்டான ஆன்சர்ஸ் பண்ணிட்டு இருக்கீங்க. The answer herpes simplex virus. The oral infection cause பண்ணதுனாலே they called as latent infections. So they called as herpes simplex virus. Okay. Which of the following is caused by herpes simplex virus 1? இதுக்கு முன்னாடி படிச்சிருந்தோம்னா இப்ப ஆன்சர் கரெக்டா பண்ணிடலாம். The herpes simplex virus என்ன டிசீஸை காஸ் பண்ணும்? The genetics இன்ஃபெக்ஷனை காஸ் பண்ணுமா, oral herpes காஸ் பண்ணுமா, chicken pox காஸ் பண்ணுமா, இல்ல cytomegalovirus various infection காஸ் பண்ணுமா? Which kind of infections caused by herpes simplex virus 1? நிறைய பேர் கரெக்டா ஆன்சர் பண்றீங்க. HRJ கீது நிறைய பேர் மெசேஜ் பண்ணிருக்கீங்க, இது ஆன்சர்ஸ் பண்ணிட்டு இருக்கீங்க. வெரி குட். The answer is oral herpes. The herpes simplex virus, they are causing an infection in oral route, so they called as oral herpes infection. Our next question. What type of genetical material is present in pox virus? Pox virus பார்த்திருப்போம். Which kind of genetical material they are presented in pox virus? Single stranded RNA, double stranded RNA, single stranded DNA, double stranded DNA. Which kind of genome they are presented in pox virus? So very good. The answer is double stranded DNA. நான் பாக்ஸ் வைரஸ் மார்க் பண்ணும்போதே சொன்னேன், the DNA they are having double stranded helical structure, so they called as double stranded DNA. So the pox virus they are having double stranded DNA structure. Our next question. The rubella is most dangerous when it's infects. இது யாருக்கு இன்ஃபெக்ட் பண்ணுச்சுன்னா யாரோட இது ரொம்ப கிரிட்டிக்கலா ரொம்ப டேஞ்சரஸா யாருக்கு இருக்கும்? சில்ட்ரன்ஸ் அண்டர் 5 years. So இண்டிவிஜுவல் இது இருக்குமா? ஸோ பிரெக்னன்ட் லேடிஸ் விமன்ஸுக்கு இருக்குமா? இல்ல அவங்களோட இம்யூனிட்டி டிப்ரெஸ் ஆனவங்களுக்கு இது அதிகமா இருக்குமா? யார டைரக்டா இந்த ருபல்லா வைரஸ் யார் எஃபெக்ட் டு வெரி டேஞ்சரஸா யாருக்கு இருக்கும்? சில்ட்ரன்ஸ்க்கு இருக்குமா? பிரெக்னன்ட் லேடிஸ் இருக்குமா? யாருக்கு இருக்கும்? So the correct answer, pregnant women. Already சொல்லியிருந்தேன். The rubella is known as German measles. German measles means they are having same character of measles. They are easily effective pregnant ladies. They are very critical and dangerous stage for pregnant women. Okay, our next question. Which of the following diagnostics technique used for direct viral genetic material? The direct virus or genetic material கண்டுபிடிக்கிறதுக்கு which test they are mainly used? Which kind of following diagnostic techniques they are used for to detect the viral genetic material? Virus or genetic material detect பண்றதுக்கு which kind of method we are used? ELISA, PCR, then heme agglutinations method, then blotting method, Western blotting methods. Which method is mainly used for to detect the viral genetic material? Okay, இதுல உங்களுக்கு சில கன்ஃபியூஷன் இருக்கலாம். நிறைய பேர் எலிசான்னு பண்ணிட்டு இருக்கீங்க. The ELISA, when we use the ELISA, here we identify antigen antibody reactions. So we identify the viral genome. So the correct answer is PCR, polymerase chain reactions. So polymerase chain reactions எங்க நடக்கும்? DNA and RNA ல தான் நடக்கும். So they called as polymerase chain reactions techniques. In the technique use பண்ணி we easily identify the viral genetical material. So இதை கொஞ்சம் கன்ஃபியூஸ் ஆயிர வேண்டாம். We detect the viral genetic material we are using polymerase chain reactions. We are using ELISA method we are using antigen antibody reactions. Okay, our next question, which diagnostics test detect for viral antigens? Which diagnostics test detects viral antigens? எது யூஸ் ஆகும்? The complement fixation test, ELISA, PCR, the human blotting method. Antigen antibody reaction we easily select ELISA, enzyme linked immunosorbent assay. Okay, here we are using 4 method: direct ELISA, indirect ELISA, sandwich ELISA, then complicative ELISA. So in the method use பண்ணி we easily identify the antigens. So the correct answer is ELISA. Okay, what is the purpose of viral culture in diagnostics virology? வைரஸை கல்டிவேட் பண்ணும்போது வைராலஜில அதோடைய பர்பஸ் என்ன? டு ஐடென்டிஃபை தி ஆன்டிஜன் ஆன்டிபாடி ரியாக்ஷன்ஸ், டு க்ரோ தி வைரஸ் ஹோஸ்ட் செல் ஃபார் ஐடென்டிஃபிகேஷன், டு டிடெக்ட் தி வைரல் RNA directly from that sample, to observe the patient immune response. What is the purpose of viral culture diagnostics in virology? வைரஸை நம்ம கல்டிவேட் பண்ணும்போது எதுக்காக கல்டிவேட் பண்றோம்? இன் அவர் டயக்னோஸ்டிக்ஸ். The correct answer is to grow the virus in that host cell for identifications. நம்ம சாம்பிளை கொண்டு போய் ஒரு செல்லுக்குள்ள கொடுக்கும்போது they are easily grow for our identifications. So it's our virus cultivation techniques. Okay, so the virus cultivation technique use பண்ணி we easily identified which kind of virus they are presented. அப்படின்னு கண்டுபிடிக்கணும்னா we are using to grow in that virus in that host cell for our identification. Okay, the serology is commonly used for to detect the viral nucleic acid, virus protein, the host antigen antibodies, then viral enzyme. The serological test யூஸ் பண்ணி நாம எதை டிடெக்ட் பண்ண முடியும்? Viral nucleic acid, viral protein, the host antigen against the virus enzyme. The correct answer, host antibodies against virus. இதுக்கு முன்னாடி கேட்டதும் இதே ஆன்சர் தான், இதுக்கு பின்னாடி கேட்டது இதே ஆன்சர் தான். Serology is nothing but antigen antibody. Serum, they are present in our blood. So the antibodies, they are present in our blood. So serum nale antigen antibody. So inge antibodies, serum, blood இப்படி கனெக்டிவ் கொடுத்துட்டோம்னா easily identify that. So the correct answer, host antibodies against that virus. Our next question, which of the following is a common symptoms for most common viral infection? ஒரு வைரஸ் இன்ஃபெக்ஷன் வரணும்னா எது காமன் இன்ஃபெக்ஷன்? ஐ ஃபீவர் அண்ட் பெட்டிக்யூ, தென் ஜாண்டிஸ், அப்டமன் பெயின், தென் ஃப்ரீக்வென்ட்லி யூரினேஷன்ஸ். எது வைரஸோட சிம்டம்ஸ், காமன் வைரல் சிம்டம்ஸ்? ஓகே, வெரி குட். நிறைய பேர் கரெக்டான ஆன்சரே பண்ணிட்டு இருக்கீங்க. இந்த ஹை ஃபீவர் அண்ட் பெட்டிக்கா இருப்பாங்க, சோர்ந்து போய் இருப்பாங்க, ரொம்ப ஃபீவரா வந்திருக்கும். they are a common symptoms for viral infection. So jaundice, abdomen pain, the frequently urination la andha virus பொறுத்து they are classified. Okay, our last question, which symptoms is commonly associated with the severe case of influenza? Pneumonia, rash, photophobia and fever. Photophobia, fever, rash, pneumonia. Influenza kuda varadu enadu? Which kind of symptoms they are associated with severe case of influenza? Rash, photophobia, fever, and pneumonia. The right answer, pneumonia. Right. So நிறைய பேர் கரெக்டான ஆன்சர்ஸ் பண்ணிட்டு இருக்கீங்க. Thank you so much. If you have any doubts na கீழே கமெண்ட்ஸ் பண்ணுங்க. டெஃபினெட்லி உங்களுக்கான ஆன்சர்ஸ் அதில் ஷேர் பண்றோம். as well as நல்லா படிங்க எக்ஸாமுக்கான. இஃப் யூ ஹேவ் எனி டவுட்ஸ் இன் எனி சப்ஜெக்ட்னா ப்ளீஸ் கமெண்ட் டு தட் வீடியோ. இந்த வீடியோவில் கமெண்ட் பண்ணுங்க. கண்டிப்பாக ஷேர் பண்ணி இதுக்கான சொல்யூஷன் நாங்கள் சீக்கிரம் சென்ட் பண்ணுவோம். ஆல் தி பெஸ்ட். தேங்க்யூ ஸோ மச்.